Okay, good evening, everybody. Welcome to the 12th and the final session of this course, Genome Editing and Engineering, NOC24BT71, which is taken by Professor Utpal Bora of IIT Guwahati. I'm Sanket Mukherjee, a Prime Minister's Research Fellow from the School of Biological Sciences, IIT Delhi. Today's session is about ethical concerns in germline editing. We will be waiting for a couple of minutes. Let everybody join and then we will begin today's session. Open meetings. Okay, let's start with the questions. Question one, the term bioethics was coined by Fritz Jar, a Protestant pastor, philosopher, and educator in Hale and der Saale in the year 1629, 1926, 1962, or 1992. I'll give you a minute to think and then answer. So the term bioethics was coined by Fritz Jar, a Protestant pastor, philosopher, and educator in Halle and der Saale in the year 1926. Fritz Jar published an article entitled Bioethics, a review of the ethical relationships of humans to animals and plants, and proposed a bioethical imperative in 1927, extending Kant's moral imperative to all forms of life. He evaluated contemporary physiological knowledge and the ethical issues surrounding the emergence of pluralistic secular society. In the process, Fritz reinterpreted moral obligations towards both human and non-human life forms by defining bioethics as an academic field, a guiding principle, and a virtue. In terms of history, uh, Charak and Susrut in 600 BC in India, they have been blessed with a magnificent code of medical ethics. The standards for a good instructor and someone who should pursue medicines are embodied in this Ayurvedic code. Additionally, it provides advice on how to interact with patients and their family members, as well as helpful hints for us to utilize when addressing topics like organ transplantation and brain death. Heinous crimes took place during World War II on captives held in military concentration camps. During this time, defenseless individuals were forcibly subjected to medical studies with questionable scientific merit. When World War II ended in 1945, the victorious Allied powers enacted the International Military Tribunal on November 19th, 1945. The first trial conducted under the Nuremberg military tribunals in 1947 became known as the Doctors' Trial, in which physicians from German Nazi Party were tried and punished for crimes against humanity for the atrocious experiments they carried out on unwilling prisoners of war. The verdict also resulted in the creation of the Nuremberg Code, a set of 10 ethical principles for human experimentation. The code aimed to protect human subjects from enduring the kind of cruelty and exploitation the prisoners endured at concentration camps. Regarding bioethics, Van Rensselaer Potter, American scientist, introduced the idea of bioethics as global ethics in his 1971 book, Bioethics: A Bridge to the Future. With his extensive and lengthy experience in cancer research, Potter presented a novel interdisciplinary idea with the aim of fusing ethics and science. He intended to create a conversation between the science of life, that is biology, and usable knowledge, philosophy, ethics, and values. And as a result, he coined the term bioethics. Potter's bioethics united humility, responsibility, and multidisciplinary and intercultural competencies. Question number 2. The Asilomar Conference was a milestone for science because it was the result of dash proposed by the scientists themselves. Is it bioethics, biosafety, self-regulation, or ethics? I'll give you a minute to think and then answer. In 1975, so Asilomar Conference on Recombinant DNA, the beginning of bioethics and biotechnology. In 1975, 140 scientists, doctors, and legislators, they gathered in the Asilomar Conference Center in State Beach, California, in order to debate the ethical implications of genetic engineering. Certain principles of biosafety were established at this conference with the object of preventing an accidental leakage of recombinant organisms which could affect human beings, animals, or the environment. The Asilomar Conference was a milestone for science because it was the result of self-regulation proposed by scientists themselves. At this conference, the scientists agreed that research with recombinant DNA should proceed, but safeguards should be outlined. In the case of heritable genome editing, there are two distinct concerns. Select the right option. One, the possibility of off-target effects of the editing process, as in the case of somatic genome editing. Two, the intended genome edits themselves might have unintended consequences, even in the absence of off-target effects. And three, some worry that taken to its extreme, germline editing could create classes of individuals defined by the quality of their engineered genome. Is it 1 and 2, 1 and 3, 2 and 3, or none of the above? I'll give you a minute to think and then we will discuss the answer. You may answer if you are sure of. This is an interactive session. We would expect answers. Okay. So ethical concerns in human gene editing. The human germline is the focus of majority of ethical debates surrounding genome editing, since any modifications which are performed would be handed down to subsequent generations. Although the discussion of genome editing is not new, it has recently attracted attention because to the finding that CRISPR may make it easier and more accurate than previous methods. However, studies that would make gene therapy safe and successful should continue, according to bioethicists and experts. Human genome editing for reproductive purposes should not currently be undertaken. The majority of stakeholders concurred that ongoing public discussion and debate are essential to letting the public determine whether or not germline modifications should be permitted. Due to ethical and security concerns, around 40 countries, including 15 in the Western Europe, prohibited or outlawed research on germline editing as of 2014. Now, talking about some ethical considerations, safety is the main priority. due to the potential for off-target consequence, that is, edits which are happening at the wrong location in the wrong gene, and mosaicism where some cells are edited while others are not. Germline genome editing should not be used for clinical reproductive purposes until it is research established as risk-free. A majority of researchers and ethicists is of the opinion that any risk cannot be justified by the potential benefit of germline genome editing. According to some experts, genome editing in embryos will not provide advantages over already available treatments like in vitro fertilization and pre-implantation genetic diagnosis. Even for therapeutic purposes, some scientists and bioethicists worry that any genome editing will pull us on a slippery slope towards using it for questionable non-therapeutic and objectives of enhancing. Others opine that once genome editing has been proven safe and effective, it should be permitted to treat hereditary disorders. And indeed, that is a moral imperative. Whatever the case, policy and regulation should be used to address issues regarding augmentation. Last but not least, some who have commented on the subject are afraid that the regulation of genome editing for reproduction would further divide various countries, leading to chaos and issues of universal acceptability. Informed consent is another thing, that people are concerned that patients of germline therapy are embryo and future generation, where it is very hard to gain informed permission. The argument against it is that parents already make a lot of choices that will impact their future offspring, even if they are equally difficult like IVF. As long as the dangers of germline therapy are unknown, scientists and bioethicists are concerned about the likelihood of obtaining proper informed permission from prospective parents. Coming to justice, as with many new technologies, there is concern that genome editing will only be accessible to the wealthy and will increase the existing disparities in access to healthcare and other interventions between the healthy and the poor, between the wealthy and the poor. Some worry that taken to its extreme, germline editing could create classes of individuals defined by the quality of their engineered genome. What are the potential risks of germline editing? So balance against the potential possible benefits of heritable genome editing are a variety of potential risks. First is the unintended consequences. The possibility of off-target effects of editing process, as in the case of somatic genome editing. Whatever standards are developed for somatic applications, there will be less tolerance for off-target effects in germline applications. The intended genome edits themselves might have unintended consequences, even in the absence of off-target effects. In case of heritable genome editing, to convert a well-understood disease-causing variant gene to a widely occurring non-pathological variant, the editing change would be to a version of the gene that is known not to have deleterious consequences. Long-term follow-up, carefully monitored clinical trial protocols would be required for heritable genome editing, with attention to monitoring off-target events as well as efficiency and correctness of the specific edit. Heritable genome editing trials would require long-term prospective follow-up studies across subsequent generations. Even those who have volunteered to be research subjects cannot be compelled to participate in long-term follow-up. Which of the following is not in the 7 principles for governance of human genome editing? Is it A, promoting well-being, B, transnational cooperation, C, respect for persons, or D, Dickey-Vicker amendment? I'll give you a minute to think and then we'll discuss the answer.
C.
Okay. So the 7 principles of governance of human genome editing is first, promoting well-being. The principle of promoting well-being supports providing benefit and preventing harm to those affected. often referred to in the bioethics literature as the principle of beneficence and non-maleficence. Transparency is the principle where it requires openness and sharing of information in ways that are accessible and understandable to stakeholders. Due care needs to be taken for patients who are enrolled in research studies and receiving clinical care. They need to be, taken care of. And responsible science, where there should be adherence to highest standards of research from bench to bedside in accordance with international and professional norms. Respect for person is also one of the principle where the principle where we should recognize that there is personal dignity of all individuals, acknowledgement of the centrality of personal choice and respect for individual decisions. All people have equal moral value regardless of their genetic qualities. Fairness is very important. Everybody should be treated fairly and equally alike. And the risks and benefits should be equitably distributed. Transnational cooperation is another principle where it supports a commitment to collaborative approaches to research and governance while respecting different cultural contexts. Question 5. The main technical problem with safety implications for potential GGE in germline genome editing, clinical germline genome editing in human embryos revealed therein include the following option except one, which is A, fairness. The principle of fairness requires that cases be treated alike and that risks and benefits should be equitably distributed. B, mosaicism, where a situation where all the cells of an embryo or organisms have the same DNA, in this case, the desired DNA modification. Off-target effects, where unintended changes in the genome outside of the targeted sequence occur, or on-target undesired modification introduced within or next to the targeted locus. I'll give you a minute to think and you can answer if you're confident. Any answers?
Sorry, sir, I did not understand the question.
Sorry?
I did not catch the question, sir. What did you ask?
Can you read the question? The main technical problems with safety implication for potential clinical germline genome editing in human embryos revealed therein include the following options. Okay, except one. So you have to choose the wrong option here. The technical problems with safety implications of potential clinical germline genome editing. Let's look at it. What are the safety issues in germline genome editing? That is GGE, as already outlined. One is mosaicism, where not all the cells of an embryo or organism have the same DNA. In this case, the desired DNA modification. There could be off-target effects where unintended changes in the genome occur at locuses which are outside where you are targeting. And there could be some on-target modifications also which are not desired, right? So these are the three main things. As per a December 2018 poll, Americans draw the line at the so-called enhancement, but favor the use of genetic engineering to address disease and A, disability, B, clinical research ethics, C, cancer, or D, oocyte procurement. Any answers?
Sir, is it disability?
You are right. So as per a December 2018 poll, Americans draw the line at so-called enhancement, but favor the use of genetic engineering to address disease and disability. Which disease and disabilities to target, however, is still an open discussion. Some question that may help inform that decision is, should there be a focus on infectious disease resistance? Should we only focus at the fatal conditions, lethal conditions? Will we decide that there is a need to quantify the degree of suffering? If an effective treatment already exists, should we still seek prevention through genetic modification? Is childhood versus adulthood onset of illness an important factor? Not all sequence variants are guaranteed to cause disease, like BRCA genes. Should they be considered? What about orphan diseases? And should certain types of disabilities be prioritized over others? These are some questions. Question 7. He Jiankui shocked the world when he broke the news that his team at Southern University of Science and Technology in Shenzhen, China, has successfully edited embryos using CRISPR-Cas9 and delivered 2 baby girls on January 25, 2018, on November 15, 2018, November 25, 2018, or December 25, 2018. If you can answer if you are sure. We have a minute till we discuss this. Any answers?
Sir, is it 25th November?
Okay. So let's talk about ethical considerations. Genome editing is a common procedure. Just give me a minute. Yeah, it's 25th November. You're right. So ethical issues of editing are not just tied to the procedure, but also the use that is made of it. The National Academy of Science and National Academy of Medicine, in conjunction with the Royal Academy of Science and Chinese Academy of Sciences, have constituted a committee on human gene editing, scientific, medical, and ethical considerations, which were chaired by Professor Alta Chero and Richard Hines, to perform in-depth study of the issue, during which input and guidance from multiple stakeholders from around the world will be solicited before issuing a consensus study report. On November 25th, He Jiankui shocked the world when he broke the news that his team at Southern University of Science and Technology in Shenzhen, China, has successfully edited embryos using CRISPR-Cas9. And apparently, he had produced twin girls who were resistant to HIV infection. But there were multiple ethical questions that came up, that how would you check this, whether these girls are resistant to HIV. You cannot introduce HIV to patients or to humans, knowing that how much it affects the life of a patient. So this is what happened. And scientists all across the globe, they came together and they condemned this action, raising the concern that gene editing technology was too premature to be used for reproductive purposes. Due to immense international pressure and in line with China's domestic law, he and his associates were convicted of violating a government ban by carrying out his own experiments on human embryos. The court declared that they acted in the pursuit of personal fame and gain and had serious medical order. He Jiankui was jailed and fined 3 million yuan, which is 430,000 US dollars or 328,000 euros. The court also handed over lower sentences to 2 men, Zhang Renli and Qin Xinzu, who were for conspiring with him to carry out these experiments. So in 1993, Chinese Ministry of Public Health releases an outline of quality controls for clinical studies of human somatic and gene therapy. In 1999, they were guiding principles of human gene therapy clinical trials were finalized. 2001, regulations of administration of agricultural genetically modified organisms and safety were published, which heavily regulates the import and domestic production of genetically modified crops. In 2003, China's Science Ministry bans the implantation of genetically modified embryos for reproductive purpose and prohibits altered embryos developing past 14 days. No punishments are attached to the regulation. In 2003, Ministry of Health and Ministry of Science and Technology jointly developed the ethical principles of research of human embryonic stem cells, which state that embryos derived by genetic modification must not be allowed to develop for more than 14 days, and that once they have been used for research, they cannot be implanted into humans or other species. In 2003, Chinese State Food and Drug Administration or National Medical Products Administration publishes the guidance for human gene therapy research and its products, which outlines requirements for applications of gene therapy clinical study. The document also outlines requirements for quality controls and product efficacy and safety results. In 2007, Ministry of Health releases measures for the ethical review of biomedical research involving humans. In 2015, Chinese researchers were first to edit genes using CRISPR in a human embryo. A gene associated with a fatal blood disorder was modified, but the embryos were not implanted. The editing was not successful in most embryos in the experiment. In 2019, Janki, who altered the DNA of human embryos, were carried to term, is censured by the Guangdong Health Ministry and fired from Southern University of Science and Technology. In 2020, there was adoption of a new civil code in China, which included personal protections for human genes and stricter regulations for gene editing. Question 8. The dash is one of the 30 genetically modified organisms to sidestep the USDA regulatory system in the past 5 years. Is it banana, BT cotton, BT brinjal, or mushroom? Any answers?
Sir, is it BT cotton?
It would be mushroom, which was approved by USA, commercialization in USA with no regulations. It would be mushrooms. Question 9. Which of the following is a safety concern related to germline gene editing? Is it improved cognitive abilities, be mosaicism, enhanced disease resistance, or increased genetic diversity? What would be the answer?
Sir, would it be mosaicism?
You're right. Mosaicism is where all the cells in an embryo, they do not carry the genetic modification. That is what mosaicism is. We have discussed about it. Name the scientist from China who edited embryos using CRISPR-Cas9 and delivered 2 baby girls in 2018. Was it He Jiankui? Was it He Muir? Was it Brokowski Muir or Muir Jiankui?
Sir, A. He Jiankui.
You're right. You're right. In 2019, which of the following organizations issued guidelines to ensure that gene therapies can be introduced in India and clinical trials for gene therapies can be performed in an ethical, scientific, and safe manner? Is it A, Indian Council of Agricultural Research, B, Indian Council of Scientific and Industrial Research, Indian Council of Medical Research, or Drugs Controller General of India?
Sir, is it C?
You're right. ICMR is the answer. In 1975, at the ACLOMA conference on recombinant DNA, certain principles of biosafety were established with a focus on A, preventing an accidental leakage of recombinant organisms which could affect human beings, animals, or the environment. B, encouraging indiscriminate use of recombinant DNA technology. C, accelerating use of recombinant DNA technology for commercialization. Or D, banning the recombinant DNA technology.
Sir, is it A?
You are right. What were the first CRISPR-edited crops to be approved for commercialization in USA with no regulation? Was it mushroom and vaxicorn, mushroom and carrots, mushroom and tomato, or vaxicorn and tomato?
So is it mushroom and tomato?
Mushroom and vaxicorn. Which of the following is wrongly matched? Is it A, 1964 Declaration of Helsinki, 1996 Dickey-Wicker Amendment, 2015 FDA approved Imlige, or 2015 FDA finalizes breakthrough therapy designation? Any answers?
D.
D is the right answer. What is Cartagena Protocol? A. Hazardous microorganisms, genetically engineered organisms or cells, prohibits germline therapy, protects the transport and use of organisms modified by biotechnology, or deregulates genetically engineered food products and processed foods.
C.
C.
You're right. Cartagena Protocol protects the transport and use of organisms modified by biotechnology, usually recombinant DNA technology. When was the Food Safety and Standards Act of India enacted? Is it in the year 2008, 2006, 2004, or 2010?
2006.
You are right. Institute for Stem Cell Biology and Regenerative Medicine use stem cells to explore a possible treatment for DASH, a common cause of blindness in India. Is it retinitis pigmentosa, hemophilia, sickle cell anemia, or Alzheimer's disease?
A.
You are right. Retinitis pigmentosa is the answer. All other diseases, hemophilia and sickle cell anemia, are blood disorders, whereas Alzheimer's is caused in the brain. So, although all of them we could potentially treat using stem cells. When did China's science ministry bans the implantation of genetically modified embryos for reproductive purposes? In 2003, 1999, 2007, or 2012?
2003.
You're right. Okay, sorry. Yeah. Okay, with this, we are at the end of this session. If there are any questions, you may ask right now. If there are any questions. So in this course, we started with the basics of, how enzymes work, restriction enzymes work. We talked about the different nucleases. We talked about FOC1, Flavobacterium ocuncoides. We talked about zinc finger nucleases. We talked about talins, a transcription activator like effector nucleases. Then we talked about CRISPR, which is the most modern form of genome editing, modern tool for genome editing. We discussed certain applications of CRISPR and the other genome editing tools in various diseases. And their potential, their applications, their potential applications, and we discussed certain concepts of which of these applications are currently in trials. And finally, on this week, we discussed about the ethical concerns, the ethics behind genome editing. So if there are no questions further, I will just stop the recording. Just give me a moment.